Greetings, glory and honor to our Lord and Savior Jesus Christ. Welcome to the Evidence Reasons Academy. I'm Salvador Cordova, and most of you know who I am. Thank you for visiting this small little channel where we explore, where we study academic things to help us become better at science especially, but also well-rounded in other fields. This is important to being able to understand and defend the Christian faith in our culture. And there's, as Hugh Ross was saying, the study of creation can help us know that there's a God and that many people can find redemption. I constantly have a television going on behind me. I have Fox News running behind me. So anyway, you can kind of know where my sympathies are, given that I watch Fox News. The category of this show is Nerds Go Clubbing. Nerds, scientists, it's kind of a tradition to have what they call journal clubs, so people or researchers will gather together and read scientific publications, often relevant to their field, so that they're able to understand their field better, and if they're publishing in it, and it's good, and also researching it, and it's good to keep abreast of what's going on. So that's how Nerds Go Clubbing. That's off of the idea of night clubbing, which is a different thing, but this is how Nerds Go Clubbing, is to do journal club readings. Today, I was researching something about, many of you know that I'm interested in multimeric proteins. There's been a noticeable silence on the evolution of multimeric proteins, and so I was just Googling around, and I wanted to see if evolutionists have had much to say about it. They haven't, except Michael Lynch. And I'm preparing for a debate November 10th with an evolutionary biologist, and I was going to challenge on the evolution of multimeric proteins. And I figured I better research a little bit of this, and so I just Googled, and so there is a little bit. I was very impressed with the paper. Don't understand it. I'm going to try to wrestle through with it. Michael Lynch has been surprisingly, even though he's dead set against creationism, he's been surprisingly insightful, and has actually been an unwitting source for criticisms of evolutionary theory on the whole. Some of the things he says, you would think John Sanford, young Earth creationist, was saying it. I sometimes laugh with my colleagues that it's like, did creationists pay these guys to say some of the things that they're saying? So this seems to be a very important paper. I don't completely understand it. People are welcome to enter in the comment section if my interpretation has been incorrect. This is a hard enough paper and challenging enough that I could get this wrong. I'll try my best though. So let's just get to it. This is doing research like this for someone who is a researcher is the cross we bear. Jesus said that he who does not bear his cross daily is not worthy of me. And so this is kind of the unglamorous side of being a professional creationist researcher and student of science. So I don't expect a lot of people to be interested in this channel. It's sort of a reality TV show, where you can just see the reality of like what life is like for a creationist researcher. Anyway, let's look at this article. The Evolutionary Diversification of Multimeric States of Proteins. June 12, 2013, PNAS, Proceedings, National Academy of Sciences. So let's just get to it. It could be some tough reading here. Let's see if I can read it this way. Hang on. See if they have a PDF. PDFs are easier. See if I can get the PDF. Let me see. Well, I'm going to try to increase the size of this. It's a little bit off, so I'm going to shrink it. Still a little off. I'll make it bigger. All right. One of the most striking features of proteins is their common assembly into multimeric structures. So for those who don't understand multimeric structures, we had the topoisomerase. You have two pieces that come together. They're identical. It's like a pair of, do I have a pair of scissors? Well, if you have a pair of scissors, that's kind of like a multimeric structure. You have two identical pieces that are joined together to make a functional machine. So multimeric proteins are very much like that. We have proteins that are either identical, really the technical term is polypeptides, that are identical, that code from one gene, that can create you make multiple copies, the multiple copies come together, and you get a functioning machine. That's what we call homomeric. Heteromeric is when you have two or more genes and they code the polypeptides, quote unquote, / proteins, and those proteins come together to make a protein complex. So the term multimeric, you have homomeric when all the proteins are the same copies of the same polypeptide. Heteromeric is when they're different polypeptides coded from different genes, or I guess there's probably a situation where they're from an alternative splice. And so you'll hear the term quaternary structure. It's the description of the details of that multimeric state. So, for example, the topoisomerase that you hear me talk about is homodimeric, meaning it's two polypeptides, identical, and they join together. That's the topoisomerase in eukaryotes. In prokaryotes, it's heterotetrameric, meaning we have a diversity. We have at least two genes coding, and the proteins, and you actually have four pieces. So the pieces of a protein complex are called subunits. We have the histones that form that are the core of the nucleosomes in humans, and the chromatin architecture are what they call octamers. Technically, you would call it octomeric. So you just have all these terms. So you have we have specific descriptions. We often use the word quaternary structure to say what kind of structure do we have. And generically you'll say, so you can either have monomeric or multimeric, and then within multimeric you have all these homomeric, heteromeric, and then within like homomeric or heteromeric, you have kind of the number associated with it. I could diagram this out. And unfortunately, people who haven't had a lot of biochemistry, this will just sound like kind of Greek. But anyway, this is why we have the academic channel. It's for there are a lot of things we get into that are not for the general audience, and that's just the cross to bear. So anyway, if you don't understand this, there are parts that I won't understand as I read this. But just for the biology students, biochemistry students out there, let's just slug through this. I think multimeric proteins will be will ultimately be not very good for evolutionary theory. Actually, a lot of things are not good for evolutionary theory, but this is going to be the reason I'm interested in this is the probability of arriving at a multimeric protein can be calculated. So let's just dive into it. And this might be just part one, reading this paper, because I'm getting really sleepy. I didn't sleep well last night, and so a little insomnia. I might have to take a brief nap shortly. Anyway, one of the most striking features of proteins, okay, this is by Michael Lynch. One of the most striking features of proteins is their common assembly into multimeric structures, usually homomers with even numbers of subunits all derived from the same generic locus. That means the same generic genetic locus. Same, did I say generic? It's genetic locus, I'm sorry. All derived from the same genetic locus. However, although substantial structural variation for orthologous proteins exists within and among major phylogenetic lineage, in striking contrast to patterns of gene structure, and genome organization, there appears to be no correlation between the level of protein structural complexity and organismal complexity. In addition, there is no evidence that protein architectural differences are driven by lineage-specific differences in selective pressures. In addition, there's no evidence that protein architectural differences are driven by lineage-specific differences in selective pressure. Here it is suggested that variation in the multimeric states of proteins can readily arise from stochastic transitions resulting from the joint processes of mutation and random genetic drift, even in the face of constant directional selection for one particular protein architecture across lineages. Under the proposed hypothesis, on a long evolutionary timescale, the numbers of transitions from monomers to dimers should approximate the numbers in the opposite direction, and similarly for transition between higher order structures. Man, this is so hard to uncoil. We almost probably have to just dive deep in it, because as I was reading this, it's like that the last 3 or 4 sentences, it's like they're not defining the concepts very well. There's a lot that's assumed, and I don't know where this paper's going. I really don't. So the abstract will probably be clearer as we go into this. So this is what I really hate about evolutionary theory. Just so many generalizations and almost, it seems like, gobbledygook reasoning. Michael Lynch is better than most. He tends to be clearer thinking. Other people are just like, I can't believe that this industry exists, because the way they conceptualize things is so poor. Michael Lynch is better, so let's see where this goes. I mean, when I say it's poor, I mean, compare, if you'll indulge me, I mean, just, compare the way evolutionary thinking goes versus, like, say, when I was doing my derivations of Maxwell's equations, the de-Alembertian leading to special relativity. This is so, in that arena, it is so precise. We can calculate the change in clock speeds based on your velocity. I mean, you would need a cesium beam atomic clock to detect it. Some of the most precise clocks that we've ever been able to build, that we can change. We actually sense the change in, I guess, even like you might say, the electron orbits. So, oh hi, Benrex. Greetings. So let me just say hi to you in the chat, just to acknowledge you. Greetings. Oh, you're not a moderator? I'm going to make you a moderator. Hang on. Let me make you a moderator so you can post. You're a moderator. You can post links. You can now post links. Okay. So, thank you. He said, a friend of mine gave me a paper which is about predicting protein structure with machine learning algorithms. My favorite sentence there is the following. Okay, so, greetings. So, I mean, this is kind of like my pride and joy for the last 3 weeks, has been these derivations I've been doing that relate Maxwell's equations to special relativity, and you don't see this level of precision in evolutionary theory. But I digress. Let's get back to the topic at hand. Okay, so this is where I'm a little aggravated. That abstract was really hard. Now, in defense of Michael Lynch, sometimes they just have limits on the number of words you could put in an abstract, and so you end up having to be just so technical no one can understand you in the abstract. Okay, so let's just read. Given the cellular basis of life, a fully synthetic theory of evolution will ultimately require an understanding of population genetic mechanisms influencing heritable changes in cell features. I'm sorry, Michael. You'll need to include physics, chemistry, and statistics. Population genetics has failed to explain this. Unless one has a better theory of modification, what's the point of talking about heritability and selection and population genetics? If you have, we see this when we just have pure random variation. If we thought random variation, and this is obvious, could work, you could supposedly you could just subject an organism to lots of radiation, a population, and if you expected complexity to emerge, it would have by now, because we could just apply radiation or any sort of chemical disruptors to the genome and it would create complexity. So they've tried to say, well then, how about selection pressure? What guarantee? No one even does a calculation. No one, hardly anyone, does any calculation of the probability a certain selection pressure will exist. They just assume it does. They practically assume that pressures to evolve things will be 100%. What if it's closer to zero? Then you're up a creek. The theory has failed. So it's not just population genetics. I mean, it's terrible. Just sorry to be on my soapbox here. And so Ben Rex is saying, within the vast space, functional proteins are extremely scarce, with estimates that range from 1 in 1 to the e to the 11th, as little as 1 is 1 e to the 77th. I'm going to modify that a little bit because I'm familiar with some of this. 1 in e to the 77th is for the beta-lactamase monomeric protein. That is a trivial protein compared to the topoisomerase homodinameric. The numbers could be even way more astronomical than that. And one thing people, I just point this out to people, you can't generalize the probability of one protein to all proteins. You can have very trivial proteins, you can have very complex proteins, like say collagen, topoisomerase, etc. And in a very, very, very rough way to judge the complexity is just by the length. So, the complexity, quote unquote, or improbability, say, of a password that's just 8 characters long, can't be generalized to a password like that, say, 1,500 characters long. So topoisomerase is like 1,500 characters long. 1,500 characters long. There's some proteins that are only 50 characters long. So, and people have been quoting Doug Axe's work where they get that figure of 1 times 10 to the 1 out of 10 to the 77th power. But that, as remote as that is that it's even more remote for something more complex. A beta-lactamase protein is about 400 amino acids, so say 400 characters long. Topoisomerases are 1,500, but it's more complicated because there's a constraint. The beta-lactamase on which that 10 to the 77th power argument is made is based on a monomeric protein. That means it functions in isolation. A homodimeric protein has 2 pieces. That means you have to have so many more connections to make it work. And I'll point out that you can, things that are monomeric, you might be able to get away with changing a lot of the characters and it will still basically function. So one thing to point out, so a beta-lactamase protein might be, say, I'm just going to say roughly 400. I know that because I studied one particular beta-lactamase protein known as the NIL-B protein for nyelinase. If it's 10 to the 77th, and if the improbability that Doug Axe gave was 10 to the 77th, and yet the protein is 400 amino acids, that means a lot of the amino acids are not absolutely critical. You could change the spelling, and I actually see that. The characteristics of a beta-lactamase, there are about 30 amino acids that have to absolutely be there. You can get a lot of flexibility in the rest and the other parts of it. The topoisomerase is not quite so forgiving, and we can also see that when we do bioinformatic analysis across species. Looks like a lot of the amino acids are critical. We also know this experimentally because topoisomerases are targets for chemotherapy, and we try to figure out ways to disrupt it. We also want to understand how it works. And my co-author, Joe DeWeese, on one topoisomerase paper, he told me that he could knock out sometimes a single amino acid and the thing's gone. So let me read what Benrex is saying. Yeah. You're welcome. He said, thanks for the moderator rights, and you're welcome. You certainly deserve it. The problem is that those functional proteins are only the functional ones, not the usable ones or the good ones. Said, thank you for the clarification. I'm aware that most trivial protein is not really representative for the minimal ones, which had to be there in the first cell. Agreed. Can we just say that the likelihood is just based on length? Shouldn't we consider multiple possible solutions in the range, but also folding and translation of it? Length is, like I said, very, very rough. Very, very rough measure. I mean, certainly, it's kind of like a password, if you might think about it. Now, we could hypothesize that there could be something where a lot of the stuff is just filler because you need spacers. Just like in mechanical devices, there are a lot of space. Sometimes you just might need just a lot of bulk space. So there are a lot of caveats to this. The important thing is, I've said the priority thing is the geometry. Just as we have parts of a car that have to be shaped right to connect to other parts, that's what I'm trying to get people to understand is think more about geometry, about connectivity. Car parts probably have tolerances within, like, say, millimeters or microns. At the molecular level, we're talking angstroms, which is kind of like nanoscale. So you have a millimeter, micrometer, and then nanometer, and then you have sub-nanometer precision. Probably, what's it, angstrom? Yeah, I think nanometer. This is. I would even guess there's some parts that have to be picometer, although I'm stretching it there. So if you need the connection of parts to be geometrically shaped and precise enough, at least within angstroms, which I think is, an angstrom is what, 10 to the minus 10th, so that's 1/10 of a nanometer. When you need to make parts that precise in order to connect, variations of the spelling that compromise that is going to compromise the part. That's why with these multimeric proteins, the protein will not function unless the pieces come together and work as a functioning whole, like the topoisomerase that is like a pair of scissors. I'm very interested because the probabilities start to become a little clearer of how likely you're going to get something that's going to work. So the old numbers of 10 to the minus 77th probability were for a single isolated, what they call a monomeric protein, where it just kind of it can function, it can catalyze and do its thing just by itself. But once we get to multimeric proteins, unless the pieces come together, a lot of times it's just not going to work. And then we're better able to characterize the statistics of how likely this is to form. And that's why this paper is of interest to me. So let's continue. I couldn't even get through the first sentence without objecting to something Lynch said, but hopefully he'll say something interesting. As much of cellular infrastructure is composed of proteins, such an enterprise should arguably begin with the evolutionary dynamics of protein structure. Remarkably, however, one of the most striking features of proteins across the tree of life is almost completely unexplored from an evolutionary perspective. Oh my goodness, I've been saying this for like the last 3 years. These guys have been totally silent on it, and there's a reason why. It's because it's bad for evolution, and he's knowing it. They're knowing it. They don't want to talk about it. They avoid the hard problems. I knew that eukaryotic evolution was a hard problem for them. I knew that multimeric protein evolution is a hard problem for it. They'll sooner talk about tiktaalik than serious problems like this where they're just open gaps. It's kind of refreshing to see someone admit this. You know, this is why I have such a hard time communicating with these guys. It's like, are you guys so blind to these problems you don't even realize it's a problem? It's plain as day to some engineers. Remarkably, however, one of the most striking features of proteins across the tree of life is almost completely unexplored from an evolutionary perspective. Well, that's probably because it shouldn't be explored through an evolutionary perspective because it's bad science anyway. Only a minority of proteins function as isolated units. Instead, most exist as symmetrical, higher-order complexes of subunits encoded by the same locus. Okay, so just if only to do I have, let's see if I have this if you'll indulge me a little bit, I think I do want to show pictures. When I was in a recent debate, I was showing oh no, I don't have the PowerPoint for that. Let me see if I can call it up. I want to show see if I could find it. I'll show some multimeric proteins. This is taking a while for this to come up. It's a little slow. Okay, so let me see. Get the display capture. Let's see if I can show it. Just echoing what Michael Lynch said. I'll show you multimeric proteins. Okay, so this is monomeric, this zinc finger protein. This is homohexameric. You can see by the different colors there. There's 6 pieces and it's symmetrical. This is a homotetrameric potassium ion channel. You can see the symmetry there. This is the homodimeric. That means 2 pieces. Topoisomerase 2 in humans. This is tetrameric, meaning 4 pieces. This is homodimeric. There are 2 pieces that are identical for the insulin receptor. Now this is heterotrimeric. You have 2 that are identical and then 1 that is by itself. This is heterotrimeric. So monomeric. So you have a monomeric here. All the rest are multimeric. And within the multimeric you have hexameric. You have the homomeric, which are these 4 and 1 heteromeric here. The homomeric you have 6, so hexameric, homohexameric. You have 4, homotetrameric. You have 2 here, so homodimeric, and you have 2 here, homodimeric. This is 3. This is heterotrimeric. And it's not easy to figure. This is where protein biology is tough to be able to elucidate that. Sometimes it's not always clear how. Sometimes it's not always clear how many parts make something up. So anyway, let me go back to the paper. Depending on the number of subunits, such complexes are referred to as homodimers, homotrimers, homotetramers, etc. So I showed examples of a homodimer, which was the topoisomerase. I don't have a homotrimer, I only had a heterotrimer, the collagen. And the homotetramers, which were the potassium ion channel. So I showed, which he was saying, what he was saying corresponded to what I was showing here. So he said homodimer, and then he said homotetramer, and this is a homotetramer. This collagen is a hetero, so I don't have a good example of a heterotrimer. Okay, so just walking through what we had. Understanding the emergence of such liaisons is a central issue in the nascent field of evolutionary cell biology. Nascent is a central issue. What does nascent mean? I actually don't know what that word means. Nascent. I should know. I'm going to have to learn something. Hang on. Nascent. Nascent. Nascent is especially a process or organization just coming into existence and beginning to display signs of future potential. I'm sorry to be cynical here and mocking. Well, maybe I'm not really so sorry. Nascent field. I think it's not a nascent field. That means it's just beginning. I think dead on arrival is probably, that's my prediction. This thing's dead on arrival. Because he's trying to frame it in terms of population genetics. You can't run away from the math, Michael Lynch. You're assuming evolution actually has a workable mechanism consistent with physics and chemistry, and that's where you're going to fail. This isn't a nascent field. I think dead on arrival is better. But let's see what you have to say. All right, so let's see what he has to say. Sorry, I can't help but gloat. I honestly saw this problematic from... This is just so reassuring, this nascent field. How about dead on arrival field? Okay, it's hard to get through even one paragraph. Understanding the emergence of such liaisons, that is, these multimeric proteins, is a central issue in the dead on arrival field of evolutionary cell biology. Most attempts to explain the existence of multimers have started with the implicit assumption that their origin and retention are a consequence of adaptive evolution, and the resultant proposed advantages are not in short supply. First, it is generally easier to find multiple small proteins than a single long one, although this does not explain the very large fraction of multimers retaining active sites within each subunit, as opposed to active sites being products of subunit interfaces. Oh boy. Oh boy, this is hard. Okay. Easier, first it is easier to fold multiple small proteins than a single long one. Yeah, like no duh. Generally easy multiple small proteins than a single long one, although this does not explain the very large fraction of multimers retaining active sites within each subunit, as opposed to active sites being products of subunit interfaces. Okay, the word interface in protein chemistry is, I'm only learning a little bit. Usually the interface is when you have, say, multiple components, and when they connect. It's kind of where they connect, or where they're close together. You said a large fractioner that way. You know, I'll just let it go. So there is, some are like that, some are not. I'm just going to move forward, see if he has a point. Second, the encounter rate of an enzyme and a small substrate is proportional to the effective radius of the enzyme. The encounter rate of an enzyme and a small substrate is proportional to the effective radius of the enzyme. I'm going to make a comment here. Enzymes are only a fraction of the proteins. Not every protein is an enzyme. Every enzyme is a protein, but the converse is not true. Collagen is a good counterexample to that. Collagen is not regarded as an enzyme. It's structural. Okay, so second, the encounter rate of an enzyme and a small substrate is proportional to the effective radius of the enzyme, and provided the catalytic site remains exposed, the elimination of extraneous protein surface may further enhance the frequency of productive encounters. Third, a smaller surface area to volume ratio may reduce a protein's vulnerability to denaturation or engagement in promiscuous interactions. The smaller surface area to volume ratio may reduce a protein's vulnerability to denaturization or engagement in promiscuous reactions. Okay, this is really thick. All right, so like if I have, basically if you take the same amount of material and you spread it out, you get more surface area per volume. But if you put it kind of like in a ball, like a globe, it's less vulnerable. What they call denaturization. It's like you're gonna, denature means you kind of remove its nature. What makes it do its, what makes it, you're removing its identity, so to speak. Removing its function, denaturing it. Even though you have all the parts there, you've kind of disassembled it. So, or you've kind of done something to it that is no longer functioning, even though you have all the parts. So that's denaturing it. Fourth, the higher order structures may reduce the sensitivity of catalytic sites to internal motions, thereby increasing substrate specificity, and oligomerization may protect otherwise unstable proteins from aggregation. He's listing all these engineering principles. These guys are just, I just want to slap them upside silly. Just because it's a good design doesn't mean it's going to be selected for before it doesn't exist. I mean, they're equating good design with selective advantage of the components before they exist. This is like, generally spark plug's not going to make, it's not going to be useful unless it's part of a system, like an engine. He's articulating what are good designs, but it doesn't necessarily make it evolvable when you don't have whole functioning systems. But we'll try to bear with it. Oh my goodness, Dr. Duff is here. Hi, Dr. Joel. Thank you for joining us. Let me just say hi. I'm going to type in a hi. Good morning, brother in Christ. He said, I've been looking at the HERK2 gene which interacts with OCA2 and causes blue eyes. The protein is huge and folds into at least 5 different regions and domain. It interacts with at least 10 other proteins. Wow. Thank you for sharing that. By the way, Dr. Joel, I haven't gotten around to reading your email that talked about speciation, and I mean I haven't been able to get through the slides, and I sincerely apologize for that. One reason I'm reading through this paper is I have a debate on Modern Day Debate with an evolutionary biologist. November 10th is coming Thursday. So I was going to put multimeric proteins on the table, and that's why I'm doing this this journal reading this Saturday morning. And I was having a little occasionally I have sleep disorder issues, and I ended up waking up really early today. So let me resume where I was on the paper. Fifth, complexation offers increased opportunities for allosteric regulation of protein activities. So allosteric regulation means a little chemical can bind to the protein complex, and you can kind of modify how it behaves. Sometimes you can get it to switch off or just not be as active, or do something else. So that's allosteric regulation. There's the formal terms for this. A biochemist can tell you what this is. So in case you missed it, I couldn't help but take a swipe here. Understanding the emergence of such liaisons is a central issue in the nascent field of evolutionary cell biology. And I was just laughing. I said DOA rather than nascent. Dead on arrival is probably a better term. Anyway, although the large pool of oligomeric structures in today's organism cannot possibly Although the large pool of oligomeric structures in today's organisms cannot possibly be strongly maladapted, this need not imply that they have arisen or are currently maintained by adaptive processes. Okay, I understand that it's so hard to be succinct. Papers have to use a lot of technical language so that it's of manageable length. So I'm going to try to uncoil what's being said. They're saying that, okay, let me just look at this. Although the large pool of oligomeric structures in today's organisms cannot be strongly maladaptive. Duh! Okay, so, the people who do protein analysis and chemistry, they know the problem of how these things, when you're trying to extract it out of a cell, with your centrifuges or whatever, and you throw detergents, you have all these proteins just kind of like clumping together in ways, you're trying to pull them apart, and it's like you quickly see the strong tendency for things to connect. It's just like because you have these proteins that have positive and negative charges, you have just like all these things that are just like, if I took, do I have it? I have some painter's tape here because I'm doing some painter's topics. So if I take some tape and inside the cell, You can make all sorts of multimeric structures just kind of randomly, I guess, if it were not well designed, because stuff would just kind of stick together. And it's like, well, if I had colored pieces or whatever, you'd really see this. If you want a really nice homomeric structure, you'd kind of like the ones that are supposed to connect together and find each other and not connect to something else. So he subtly said that there's an interesting problem here why you have these oligomeric. Oligomer meaning small number, and sometimes the term oligomer is like less than 50 units. I saw that in some biochem book. That's totally arbitrary. So small number like say less than 50, maybe less, arbitrarily I'm just going to say less than 10. If you have like anything less than 10 parts and it's all homomeric, you want them to find each other in that cellular soup, and it's being translated. They're going to find each other and just join together nicely and work. And they're saying, isn't that amazing that most of the let's see what it says here. The large pool of oligomeric structures in today's organism cannot be strongly maladaptive. Why don't they just use more straightforward language? I would have said this. It's very clear that even though polypeptides might have a strong tendency to just connect to each other randomly, they don't. And the ones that, when they do connect, they're highly functional. Evolutionists keep using non-design language, all this stuff about maladaptation. They need to use engineering language. It'll make more sense. This need not apply that they have arisen or are currently maintained by adaptive processes. So he's saying it didn't have to arise because it was useful immediately to the organism, or that it's maintained now because it's useful to the organism. I'm like, okay. And Lynch is one of the better writers, so let's keep going. Indeed, despite the plausibility of many of the above hypotheses, empirical evidence for the adaptive value of alternative multimeric structures is essentially completely lacking. Let me just read that again. I'm sorry to laugh. Sorry to gloat here. Indeed, despite the plausibility of many of the above hypotheses, this is my problem. There is an abundance of hypotheses, not a lot of empirical evidence. What else is new? Indeed, despite the plausibility. No, it's not plausible. He could strike that. Indeed, despite the many above hypotheses. Don't call it plausible. That's an assumption. Empirical evidence for adaptive value of alternative multimeric structures is completely lacking, and a number of examples can be pointed to in which a more complex structure seemingly operates no more efficiently in its lineage than a simpler structure in others. Oh my goodness. Oh my goodness, this is terrible. I mean, I cannot believe this. Do you guys ever study stuff in detail? This is blown apart so easily. Okay, yes, you have heterotetrameric. You have helicases and you have hexameric. So tetrameric being 4, hexameric being 6. So you do have variety there. But do you really think, and I'll show you this, going back to this protein orchard, I mean, Mike, there's certain things in terms of physics you should consider rather than evolutionary biology, and I think the picture would be clearer for you if you would just kind of put your thinking hat on. Let me see if I can get this going here. All right. Why would this topoisomerase, why would you think it would be trimeric? This is not gonna work. This is like a pair of scissors. There's a reason they're just 2 pieces. Good gravy. Now, for this one, this is a tetrameric structure for a potassium ion channel. That little hole in the middle has to be precise within sub-angstroms, which is like fractions of a nanometer. This just doesn't work. Dude, there's reasons geometrically that it's optimized to be, if you would be willing to accept design, you compromise the geometric viability of it if you're just going to, haphazardly say, oh, let's reconnect it, go from a dimer to a tetramer. There are just issues with that. Okay, so we have topoisomerase in eukaryotes, it's 2, it's a homodimer. And prokaryotes, it's a tetramer. But you need to retain the symmetry. You're not going to go from like a dimer to a trimer. Even the tetramer has a certain scissor-like quality where the 2 sides, the 2 parts in that hetero tetramer are still like scissor-like, and you can kind of see this. Let me grab. Okay. In the prokaryotic gyrase, you see this is a pair of tongs. Just like, let's say it's like the topoisomerase. That's the scissors. You could see there's one part here. You could see there's kind of the handle part and then kind of the grabbing part. All right. And then, so you look at the topoisomerase, you can kind of see like it has an upper part and then a lower part, just like these tongs. So for this cannot be haphazard. Mike, this cannot be haphazard. It's like, maybe it's adaptive or not. It's like very, it's going to be maladaptive. It's very easy for it to be broken. It can't just arise willy-nilly. Okay, let's go on. He's confirming a lot of what I've been trying to say. Anyway. Oh, this is painful reading. This is why I hate reading evolutionary literature. This seriously. Look, we have many of the above hypotheses. Empirical evidence for the adaptive value is essentially completely lacking. Many hypotheses, empirical evidence completely lacking. That's evolutionary biology. That's why I resent reading. I just go nuts. Not only is it completely lacking, there's a lot of evidence against it. But let's go on. I just go nuts reading this. It is sickening. Did a creationist pay Michael Lynch to write this? It's great. So this is why I really like Michael Lynch's stuff, because he'll say stuff like that, but he'll be brutally honest. Empirical evidence completely lacking. I love it. So even though the rest of his stuff I object to, he'll get to the point I'm looking for. So let's go on. Empirical evidence is essentially completely lacking. Boy, there's lots of stuff here worth quoting. And a number of examples can be pointed to in which my more complex structure seemingly operates no more efficiently in its lineage than a simpler structure in others. I mean, look, like the topoisomerase. Why are you going to have like 10 pieces? You know, it works well with the 2 pieces that are put together. There's no reason to have multiple. It works. There's an optimal number. Drives me nuts. First, to achieve a critical concentration of an active multimer, the expression of monomeric subunits must be raised to a high enough level to ensure an adequate number of encounters for successful complex assembly. You only need 2. Well, yeah, concentration's important, but you know the more important thing, and I'll tell you the more important thing, the more important consideration. See if I have it here in my, okay, I had the orchard. Let's see, did I have it here? I'll show you the far more important thing. And this ties into the probability. See if I have the diagram from Bruce Albert's Essential Cell Biology. Where is it? Here it is. This is like the money diagram right here. Let's see. I'm trying to bring it up. Hang on, guys. Let's see. It's more than just concentration. When you have a multimeric complex, so like you have 2 polypeptides / proteins, they need complementary geometric shapes. And the geometric shapes also often have charge distributions. You have negative in one, positive on the other in the right location, and then they'll come nicely together. They can find each other in that random Brownian soup and spontaneously connect to each other. I got to emphasize, this is just nuts, because I mean, see, I have these sticky parts here. They could connect in all sorts of different ways. You know, if I just spontaneously just kind of throw them together, they could connect. It's very easy to connect in the wrong way. These charge distributions are so beautiful that you could just throw it in solution. They've done this with the topoisomerase, and I'm sure with others. They'll spontaneously come together. Now, there are times you need some sort of chaperoning process to help it along, to get them to connect. But really, this is just amazing that you have all this random stuff going on. And I'd say protein concentration is the least of your problems. It's being able to get the right charge distribution. Not the right charge, you need the right shape so that they can present themselves to each other and connect, and then you have the charge distribution. And this is all done through spelling. So, I mean, I would like to ask a protein chemist, can you make a homodimer for me? I think the least of his problems would be the expression level. Make a homodimer that will stably connect, or a hexamer, or whatever. How about, okay, so let's say the problem of making a potassium ion channel. You have to have that little hole. Let me see if I can show, where did I have it? Let's look at the potassium ion channel again. You need to have a little hole here that is sub-angstrom precision. Why don't I ask a protein chemist and say, why don't you just kind of make it a little bigger? Like, how about 0.2 angstroms bigger? Write spell out a polypeptide that when you make 4 copies of it, it's going to spontaneously come together and be about 2 angstroms bigger. Just try to do that. I mean, just think about how hard that is. I mean, you could, you know. I mean, look, we can. It doesn't even have to be a tetramer. Make it some sort of multimeric protein, because we have here, like the hexamer, this homohexameric helicase. It has a hole there that has to be about the right size so DNA can go right through it. Single-stranded DNA will go through that hole. A potassium ion will go through that hole. So it's possible to make all of these multimers with a hole in it. Like this one has a hole of a certain size. This one has a hole of this size. And the topoisomerase has a hole of that size. So he's talking about the complexity, the difficulty of having enough concentration. I'm just like, I think you're straining at a gnat and letting a camel through. Let's see if he's going to even mention the difficulty of geometry and charge distribution. Anyway, oh boy, let's just keep going here. This increase in subunit production will entail an energetic cost. Second, unless a newly emerging dimer has a synthetic symmetric interface and the correct subunit orientation, concatenations into indefinite filaments can arise. Yeah, no duh. Concatenations to indefinite filaments can arise. Okay. All right, I'm going to use my tape here. And so, yes, you see the problem? You could have all these sticky things, and you could just keep getting it. This was actually really a smart insight. One of the best insights I've seen in the paper. I get it. I mean, what'll happen if you just keep sticking all these things together? I mean, look, let's just look at this. I mean, this is for once he had a really good sentence right there. Because this would be really bad if you had indefinite, like he said, I liked his wording. The indefinite, he said, indefinite filaments can arise. Let me just highlight that. Unless a newly emerging has a symmetric interface and the correct subunit orientation, concatenations into indefinite filaments can arise. So let's look what might happen if we didn't build this correctly and you get an indefinite. Did he use the word? Okay, you get an indefinite filaments. So let's look what this means. So let me bring up the orchard picture again. So what would an indefinite filament be? So let's say you're trying to build this insulin receptor here, and you just kept getting copies of this left-handed side, and it's just going to keep attaching itself. I mean, this is not trivial. So what Bruce Alberts actually pointed out is you want to have certain kinds of amino acids on the outside and certain kinds on the inside, and he called that kind of like a pressure. It's in essential cell biology. So the idea is, well, just kind of like the way you have the tape, but it's a special kind. It won't connect one way. It'll tend to connect very nicely in a certain way. So like you have, like the way I just connected the tape, you don't have the sticky side on the outside when I connect it. I mean, this is not the nicest way of describing it, but this will spontaneously happen, that it's going to come together, and the parts that are supposed to attract will attract. And that is really interesting in the way that this homodimer is constructed. I mean, it takes a lot of foresight to construct it. If you're trying to, I mean, just to think how hard it is to build something that will, just a simple dimer the way it's spelled, that you have a, in 3 dimensions. I mean, just try doing it one time. Just try doing it one time. If you build I don't know quite how to describe this. This would be a very good exercise for someone, even without doing all the mechanics of the protein folding. Let's just say you were laying out the spelling on some arbitrary let me just even try something like a Kleenex box, and you could put just letters here, or just pluses and minuses, and you need to write the pluses and minuses so that when it wraps around in the shape, it's going to be able to give you that charge distribution so that it will connect a certain way. And then on the outside you have kind of neutral, uncharged amino acids. I know I'm not being very clear, but I think if I had a classroom and I gave people an exercise to write it, you could see it's, at the very least, you could see you need some level of design. It's very hard to get this to happen randomly, where you want certain amino acids to be on one side of the polypeptide, and you want charged amino acids to be on the other side, but the charged ones have to be positioned so you have plus here, and then on the corresponding part, you're going to have the charges plus and minus connect. Because like if you have plus and plus side by side, and then you're trying to get these to come together, they're not. Now you may want to do that to help shepherd it. I mean, this is like not trivial. And Michael Lynch, again, every now and then there's stuff that he writes that just like, ugh. But then he'll make a pointed statement and an important concession that this, how does this happen? I'm just like, yeah, that's exactly it. He called it. In fact, that was pretty insightful. That was worth it right there. When he was saying, unless a newly emerging dimer has a symmetric interface and the correct subunit orientation, concatenations into indefinite filaments can arise. Yeah, like no duh. Numerous human disorders are known to result from inappropriate protein aggregation. Yes, yes, you got it, baby. That's right. This is really hard to do. Inappropriate protein aggregation. You can't just, this is why you can't just mutate. You know, you can't just have a gene duplication and think it's going to just be happily along. You can create these inappropriate, and inappropriate is such a teleological word. Inappropriate. It's like, there's good design and bad design. I like that. Inappropriate protein aggregation. And highly expressed proteins are especially vulnerable to promiscuous interactions. Oh my goodness. Promiscuous interactions. Um, well, one way that you don't get promiscuous interactions when you're assembling a car is all the parts are shaped to connect with only a specific other part. A promiscuous interaction is when you get one part that's connecting where it shouldn't. So, particularly in electrical engineering, all your wires can be shaped about the same. And they color code the wires so that when you're assembling it, if you have to do it manually, you try to connect the right colored wire to the right other colored wire. So hopefully, that's why when you're just trying to lengthen something, you want to connect white to white and red to red. You don't. Otherwise, you could generate short circuits. So that would be kind of the analogy of a promiscuous interaction. So there are. It's really nice, So if you have a promiscuous interaction, if you get something that, like let's say you have an enzyme, and the wrong thing gets inside of it, and it shuts the enzyme down. I forgot there's a certain term for that. I'm open, Dr. Joel, if you could find something better, but I don't think it's going to work. The more that I hear about some of what they're doing or hypothesizing, the crazier it sounds to me. So you're invited to bring a more up-to-date. But, that's a fair criticism that this is 10 years. But I don't foresee it because they're fighting basic statistics here. This cannot be, doing more evolution is, I think it's going to crumble under the weight of physics and chemistry. But anyway, third, in diploid species, some dimerizing mutations may have deleterious effects in heterozygous individuals as a consequence of heterotypic complexation, a process that can greatly reduce the probability of establishment of the dimerizing allele. Okay. Third, Benrex says, color coding is not always easy. I had a 40-wire color code cable. Okay, so let's go here. Third, in diploid species, some dimerizing mutations may have deleterious effects in heterozygous individuals as a consequence of heterotypic complexation, a process that can greatly reduce the probability of establishment of the dimerizing allele. Let me just highlight the sentence here. I gotta credit Michael Lynch. He certainly, he's listed about 10 references, so some people have been thinking about this. I just have to laugh. It's like a number of hypotheses, but empirical evidence completely lacking. Actually, there's a lot of empirical evidence against it, so you need to be a little bit more honest here, and he's actually providing some evidence negative to all of these hypotheses. It's not that it's lacking. It's more accurate to say most of the empirical evidence is against any of these hypotheses. It's not, as I've said, the evolutionary, the emergence of complexity is not consistent with what we know of physics, chemistry, statistics, geometric connections. This is intuitively, this is why I like multimeric proteins. The problem of geometric specificity is just there in your face. And so this is a great passage right here. In diploid species, some dimerizing mutations may have deleterious effects. Okay, so I'm going to try to uncoil this. So you have a diplate species, so like humans, male and female. So you could have one gene, and then you have a gene locus, and then you have alleles, meaning variations of that basic gene. So like, you have the chromosomes that are paired, and you have the gene, presumably the gene locations. They call it locuses. I just call it the gene locations. Okay, the gene locations corresponding on each, and each, it's the same location, but they're slightly different spellings. So the gene on one chromosome is spelled differently on another. So when you have that variation, and then it can be causing what they call he calls the dimerization, and I never even thought about this, is that, and I need to ask Joe because he is a specialist in this. It never occurred to me. Let me just call this out here. It's not just dimerization. How about tetramerization or hexamerization? Let's look at this. This is an interesting thing that Michael Lynch brought up. There is the possibility. Okay, let's just say we have alleles for like a topoisomerase here. This one could be one thing, and on the left it's one spelling, on the right it's a slightly different spelling. Or in the homohexameric helicase, you could have a whole mix. What do we do then? Or what do we do here? So Lynch isn't talking about that scenario where you have an existing functional dimer. He's talking about something that was monomeric. Monomeric being like the beta-lactamase or like this zinc finger. And then now you have a mutation that's going to try to dimerize this. Oh my goodness. This is why I like reading Michael Lynch. He has a critical mind, but he can't quite see that his theory's blowing apart, but he's honest. He thinks. Oh, Schnoil is here. Greetings, Schnoil. Good morning. God bless you. Well, it's good morning here. I'll fill you in. I'll fill you in here, Schnoil. I was preparing for my debate. That's an announcement. I'm going to debate an evolutionary biologist on modern day debate, just 1 versus 1. And 1 versus 1 this coming Thursday. And I was going to confront him on multimeric proteins. So multimeric protein, just using a prop here, is like the topoisomerase. You have two pieces that are identical, and then you stitch them together and you have a working complex. You see, you need a little foresight to make something that'll actually work when it's in the context. Because, what can I do with this if it doesn't connect? If I just have one piece, maybe I could use it as a club or something, but it makes more sense in a functional context when it's two pieces. So there has to be foresight. But in the context of a cellular environment, a lot of these things self-assemble spontaneously. We've demonstrated, at least for topoisomerase, you could throw the pieces into solution and it's going to form these scissor-like things naturally. That's just amazing because, here in the macroscopic world, we have to put little fasteners and screws to put things together. These things come together spontaneously. It's just like, throwing parts in a bag, shaking it, and then out comes all these working machines. The way that can happen is there are charge distributions that enable this magical process to happen, and I want to show a picture of that charge distribution. So yeah, let me copy this. This is too good. This is really good. So let me put it here, because this is such an essential concept. So to have the parts come together nicely, they have to be geometrically shaped. But then you could see the little dots there. The red is the positive and the blue is the negative. And so it has to have charge distributions to enable it to spontaneously connect. And then you could see on the outside, this is a little subtle, is there isn't so much charge on the outside. So certain amino acids will, where they are, might make them more negative and others more positive. And then you might have something neutral on the exterior. And so this enables spontaneous assembly of these molecular machines. So it's out there in solution, and it's going to find its partner. And importantly, it doesn't partner, preferably doesn't partner with the wrong partner. See, and I was giving the analogy, like if I had tape, these little balls of tape, and I would just throwing them into a box, and let's say you wanted to have only certain shapes connect to each other, but if they're very sticky all over, they would just, you get connections that would be undesirable from a functional standpoint. And so these things have to, one, you have to be able to find and connect with each other, but it's also desirable you don't create something that is just going to go around and connect to everything and start jamming up the machinery and preventing connections. You just have a lot of useless junk floating around. So this is very problematic, and it gets even worse because when you have diploid species like humans, where you have pairs of chromosomes, like mother and father, and there's all the, there's the exchange of genetic material. What'll happen, like say, if you inherited from the mother one kind of, you have ideally, like for something symmetric, what they call a dimer, these are identical. But what if the mother has one version of one of the scissor and the father the other half of the scissor? I realized something that could compromise the operation, and it never occurred to me. This is a really big problem. That could compromise it. Now, Michael Lynch wasn't referring to that. What he was talking about was, what if you start out with just one thing, and then suddenly there are mutations to make it to start to change the function to be a multiple component. So, like, I guess for a pair of scissors, you could get it kind of be like a knife. So let's say you started out with a knife, and then somehow you figured out to make it into a pair of scissors. He's talking about the complication of how that will happen, and that is this part here. He's talking about it here. In diploid species, some dimerizing mutations may have deleterious effect in heterozygous individuals as a consequence of heterotypic complexation, a process that can greatly reduce the probability of establishment of the dimerizing allele. Oh my goodness. That was unbelievably insightful and shows a lot of other problems. So let's say you have, let's say it was an enzymatic process. or and it works so well as a single molecule. It works so well as a single molecule. Now you have something that will just make it connect to something else. What we call dimerizing. Let's just imagine you had a knife that you're trying to make a pair of scissors. What happens if we get a mutation that will just make it two, but then it can't ever open up? It'll just stick together really, really well. Well, that would be selectively disadvantageous, because you've just made a mutation that's like, well, yeah, you made something that normally is just hanging around one by itself, but now it's two, but then in the process of making it two, you have shut down its function. Again, geometry comes and takes a bite out of evolutionary theory. There's an essay by Joe Felsenstein, Mathematics versus Evolution. I don't think he realized how good a title that was. Because in mathematics, there's lots of geometry. And I don't think, they're coming to terms with it. So Michael Lynch was saying this is kind of a burgeon, a beginning, kind of an infant field of evolutionary cell biology. I wanted to say it's more like dead on arrival. I mean, he's giving me all the reasons right here, many of the reasons why it's dead on arrival. It just doesn't square with mathematics and geometry. I'm even more convinced now that I'm right. Told you so. And this is really great. I mean, this raises an interesting question. Let's see, diploid species. I have to call up my Uniprot. They have the spelling of the proteins for like humans and the spelling of the proteins for, let's see, some like Arabidopsis thaliana of the same homolog. Why aren't the alleles variable? This is a good question. If they keep saying in diploid, oh my goodness, in diploid species, the evolutionary biologists, this is why I like Michael Lynch, because he really thinks hard about these things, and he's in contact with people that really think. So you have kind of the evolutionary biologists, all they do is just phylogenetic analysis. They never think about the problems of their theory. Then you have people like Michael Lynch who's in touch with people that think deeply about this. So let's look at all the multimeric proteins. And you notice they'll say, oh, look at this evolutionary clock. This evolutionary clock, it just easily diversified. How come there are not more alleles for these dimeric proteins? If it were, if there weren't selection, or if it weren't essential. This is indicating that these species arose from a recent common ancestor recently. That never occurred to me. Oh my goodness, this is devastating. Oh my goodness. Oh my goodness. I wish Emory were done with grad school. We would have a field day with this. I need to talk to some bioinformatician who's a young earth creationist too, which there are not many. Let me show you what I have in mind. So let me bring it up on my screen. This has been already a great nerd session. Let's see. Cut this one down, and I'm going to go to Uniprot. And I'm going to look up Oh man, they changed the interface. I like the table. So we have the topoisomerase in humans. Oh, we could have a field day. So we have the spelling here. Boy, they changed the interface. They changed this. I can't read this anymore. What did they do? Oh, look at all those variants. Oh my goodness. Here are the alleles right here. And a lot of them have disease association. Oh my goodness, look at all those alleles. Ooh, yikes. I'm looking for a spelling. I don't know how to navigate this anymore. They changed the interface. Okay, I'm going to read Dr. Joel's said, simple, not more alleles because of select. See, there's the problem, Dr. Joel. There's limits to how much selection can work. That's the problem of Muller's limit. There's a mutational load associated, and we don't know how well that's even in operation, and that's assuming selection actually works. I mean, the strength of we have experimental evidence, particularly with certainly with histones that are deeply conserved evolutionarily. They did histone deletion mutants, and there's no evidence that there was strong selection, which is kind of very disturbing. So there's a lot of deconservation where the segments are removed or altered, and there's no effect. No immediate effect. I believe it's functional compromise, but this is still very bad, because the selections at play that you have to mutate both alleles simultaneously so that you maintain it. So Arabidopsis thaliana will have one set that's very close in similarity among, and it's diploid, or at least possibly polyploid, and then you have something like the mammalians. And why are the alleles moving in lockstep with each other? So let's assume selection. This means that a lot of the positions are actually optimized, which is contrary to a neutral clock. As always, the evolutionary biologists never really think hard. I'm sorry to be mean. Except for people like Michael Lynch and his colleagues. They're really thinking about this. And it just occurred to me with multimeric, not just dimeric, you can say trimeric, tetrameric. I don't know of many pentameric, hexameric. How are you able to maintain the alleles within a species are so similar compared to so you have one set of alleles for humans, different set of alleles for Arabidopsis thaliana, but within Arabidopsis thaliana, the proteins are kind of like, you could take one allele in an Arabidopsis and know that it's very different from a human. But the alleles within the human set are all very similar to each other relative to like Arabidopsis thaliana. And the alleles in Arabidopsis are very similar to each other relative to, like, say, the human, the human form. How is that accounted for? Except by recent special creation, special recent creation. That would make the most sense. I never even thought of that. Multimeric proteins, I had sensed it for a long time, are going to be problematic for evolution. I just want to slap creationists silly and say, why are you so obsessed about human chimp ancestry and those bones? You're getting it handed to you where the evolutionary biologists are practically telling you empirical evidence is lacking. Many hypotheses, empirical evidence completely lacking. And I would say it's not even that good. The empirical evidence is against it. Each of the hypotheses. Ugh. Why do these creationists keep talking about human chimp ancestry? You got it handed to you on a platter. Oh my goodness. Michael Lynch has handed it to us on a platter. Schnoil said, if it's available next semester, I'll take a bioinformatics module. Oh, praise God. It's not, okay, I know, for the grad students out there like Ben Rex, Mr. Jetty, and Schnoil, I could tell you, because I've taken a lot of you guys' classes, you can learn this. It's not that hard. I'm a retread. I'm not really a biologist, and I'm, it is not, the thing in computer science and programming, so much of what you learn has to be dependent on what you learned before. Whereas for a lot of the biology you need for evolution, to criticize evolution, you can learn a lot of it really, really fast. So you have to learn, like, proteins are made of amino acids. It's kind of helpful to know the names of the amino acids. And you have to learn some terminology like, quaternary structure, and then kind of some of the genetics, like, haploid and diploid. And you could start, you'll have enough to have a critical eye to see what's not being explained to you. So thank you for joining us, Ben Rex. Let me just say goodbye to you here. God bless you. And Schnoil said it's like it was 2 p.m., so it's about 3:30 p.m. where you are. I just happened to be up early this morning. Bioinformatics, I think you can do, because a lot of it is string processing, string storage, and string comparison. So you, as a computer scientist, Schnoil, you've done string processing, you've done string comparison, string storage. That's bioinformatics. Seriously, how is it radically different from any other database that stores strings? And then they'll add more data to it, like, the experiments involved in deducing this or whatever, and you just have these huge databases of strings. I know that may sound, it's, there are algorithms, because it's not, you don't have a lot of exact text string searches, you have a lot of approximate searches. So like the spelling of a protein in a human, like again, like topoisomerase, the homolog, that means the corresponding kind of architecture spelling of that architecture in another creature. It'll be spelled differently, but it'll have the same characteristics. Just like the parts of a car you can recognize, like a steering wheel and spark plugs and all, but they will be shaped, they'll have characteristics that are a little bit different, but you would still recognize it as the same thing, like a tire, a battery, and that's what kind of homologs are for proteins, and they're different across species. So whether, I don't know if they offer bioinformatics where you are, but I think if you look at it as basically storing strings and comparing them, it's very similar to what you work with databases. The only thing is that you don't have exact matches. They're algorithms that can look at strings and say this is the same kind of thing. So just like your eye can recognize a spark plug from, even though they're different across cars, you could still recognize it has the same architecture. And these strings will have defined architectures. The visually showing it, and they're very few that you can actually visually see that something has the same architecture, but I have some examples of that, and I'll show you. These are my favorite. This is why I like these. You can visually see that they have the same architecture. Let me see here. So on the left is a collagen, on the right is a zinc finger, and I colorized the string where here all the G's are colored red, and let's see here, do I have it? So you could see, even though these two strings are different, they're 60% similar, so they have, which means they do have a lot of variation, but you could see they have the same basic architecture. I colorized the C's here, and I did have to put a little spacer in here to get it to align. So that's kind of like the only difference. The databases are not exact. And Dr. Joel says bioinformatics courses can be all informatics and little biology or more biology. Will depend on which department it is taught from and who the clientele. Thank you, Dr. Joel. Thank you very much for that comment. So maybe kind of when you could see it, that like when I'm doing a text search or spelling, sometimes the computer can kind of say, I think you wanted this word, you misspelled it, but you're close enough. And so there's a lot of, it's a little more complex than that, obviously, for detecting homologs across species, but there's still kind of the sense that we call kind of the distance between the strings. Like a single misspelling, you would say it doesn't have a lot of distance from, a single letter misspelling of a long word means it's not very distant from the correct spelling. And then as you add more characters that are wrong, it's more distant. So you kind of have the idea of distance between strings, and then you can group these strings together, being that they're all kind of close in similarity, and this is what they call homologs. PCA, what does Schnoil said PCA? A lot of the algorithms I came across are used heavily in bioinformatics. I've never heard of PCA. Anyway, you don't have to spell it out. So thank you for sharing that. So this has been a very fruitful reading of a paper. It's getting me a lot to think about. And let's just see what he has to say here. A case, he says results. How can he have results? So did they have experiments? A case for stochastic transitions among oligomeric states. Approximately 2/3 of the protein for which well-curated structural data exists are known to assemble as homomers. Okay. So that doesn't mean all proteins, but proteins we have. Physical component analysis, PCA, thank you. So proteins that we have a lot of data on, what they call well-curated, and it's high quality, well-curated, and structural data exist are known to assemble as homomers. Moreover, although imbalances across protein families and or organisms presumably cause biases in the current databases, the distribution of multimeric complexity, number of subunits, are quite similar across phylogenetic lineages. Roughly 2/3 of multimers are dimers, 15% are tetramers, and the remaining classes exhibit progressively lower frequencies. Oddmers. I want to see this. Oddmers. I like that term, an oddmer. That'd be oddmers are noticeably underrepresented, with, for example, trimers having less than half of the frequency of tetramers. Oddmers. Okay, so let's go back to my diagram. I used the word oddmer. So I guess the counterpart of that would be evenmer, the evenmers, even numbers. So let's go up here. Oddmer and evenmer. Schnoil says, we also went through a few clustering algorithms which I've seen being used by creationists trying to distinguish between kinds. And I'm going to get myself in trouble here. I don't like baraminology. There are too many unknowns right now to be... Why are you worrying about that? That's okay. I can thank God for the baraminologist because it makes me unique, more unique among creationists, because I'm doing anti-evolutionism here. Now, these clustering algorithms work really well for making protein orchards. In fact, did I have to make this? No, the clustering algorithms that are in mainstream databases automatically do all the clustering for you. You don't have to be a baraminologist. So, that's what I try to say. Why are they focusing on trying to look at created kinds? You're having it served to you on a silver platter for the independent architectures of the protein families. But back to the point. So most of the multimeric proteins are even. So this is 6 hexameric, this is 4 tetrameric, this is 2 dimeric, and then this is 2 dimeric. We have a tetramer here, but that's not homo. It's a mix. They're not all the same. Okay, so let's go back to the oddmers. That is, oddmers would be 3, 5, 7, 9, 11. Those would be oddmers. So 3 being trimeric, 5 being pentameric, 7 septameric. I don't know what you'd have for 9 or 11. Just 9-meric, 11-meric, 13-meric. Those are rare. You have a lot of 2, 4, 6, 8. 2 are the dimeric variety. this is the first one of the few evolutionary papers I really am enjoying reading. And I probably need to take a nap now because I just had disrupted sleep. That's why I'm up. A less biased picture of the diversity of multimeric structures can achieve by focusing on specific sets of proteins with function conserved across the tree of life. The enzymes involved in glycolysis of citric acid cycle comprise two such groups for which substantial structural data exists. For these pathways, nearly every protein that has been studied in more than a few major lineages exhibit two or more oligomeric states, with variation often existing both within and among major lineages. Multimers for these proteins are almost invariably homomeric rather than heteromeric, with subunits encoded by different loci, although there are certainly plenty of the latter for other loci in all domains of life. Moreover, in accordance with the broader analysis noted above, there is no association between the level of multimeric complexity and organismal complexity. Okay, I totally know where he's going with this. He's talking about the TCA, the Krebs cycle, citric acid cycle. I mean, these are the same thing by different names. Krebs cycle, citric acid cycle, TCA cycle. And there was, I'm going to embarrass myself. I used to know the names of all the proteins in this pathway. So let me just show a picture of this pathway, that what he's talking about, the citric acid cycle. And there's this protein called isocitrate dehydrogenase or something like that. That's the one that I remember. Schnoa says, I'm not convinced by any methodology regarding baryomenology. I don't really see this as helping our case at the moment. I agree. Oh my goodness. In fact, it ends up being a distraction. Aaron Ross says, well, so what are the created kinds if you're going to insist on it? You baryomenologists can't figure it out. And just like, see, that's exactly why I don't feel it helping our case that much. Maybe it'll help you if you figure out how many animals can fit on the ark. I mean, I get that, but okay, and some of my friends are baryomenologists. I mean, I think in time we'll be able to, it might be useful, but how does it help us? Why try to build these models of created kinds when we need to fight evolution and find out how wrong it is? And I don't think we're really good at that. We're getting better, but that's just my opinion. So we're of kindred spirits on that topic. So let's look at the citric acid cycle here. And so isocitrate dehydrogenase. That's the only, I don't know why that just sounded like such a cool name. Here, isocitrate dehydrogenase. So you have acotinase, isocitrate dehydrogenase, alpha ketoglutarate dehydrogenase. So a dehydrogenase means it removes a hydrogen, I guess. You have succinyl CoA synthase. For a final exam, professor said, you will recapitulate this on the final exam. You'll be tested if you can draw that diagram, put all the labels, put all the chemicals, put all, etc. And it took me I had to spend hours just kind of, I'd start to draw the circle and I'd start to put the names of things and I'd forget and I'd go back to the diagram. It's just like memorizing scripture, just a lot of rote memorization in biochemistry. So what they're saying is these enzymes are multimeric, and across species they vary. So let's see, isocitrate, let's look at a picture of it. And Ann Gauger was pointing out that, let's see, quaternary structure. I don't see one. There are, Ann Gauger was pointing out, she works with the Discovery Institute. There was one enzyme that had like 60 components, but that's only true in one species. And I'm trying to see. Ann Gauger. Ann Gauger. Let's see if it was a dehydrogenase. Okay, Sandwalk, a creationist. Anyway, I'm going to go back to Michael Lynch's paper. There are see if I get one with 60 parts. I'm looking for one that has a lot. There are some that have a huge number. And one of them was with, you see, too small. I'm looking at pictures of this. Yeah, I'll show my screen here so you could see what I'm agonizing over. I'm trying to find one with a lot of parts. Oh, there's one. Inflammasome. Now the inflammasome. That's an interesting one. An inflammasome. Oh, what's this one? Look at that guy. It has 1, 2, 3, 4. Let me count it. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. NLRP3 is the name of the protein. So I think when you have lots of them like that, you can get variations in the number. But anyway, I'm about ready to wind this down because I'm getting kind of tired. So let's see what Michael Lynch has to say. So he's going to cite this. The enzymes involved in glycolysis and citric acid cycle comprise two such groups for which substantial structural data exist. For these pathways, nearly every protein that has been studied in more than a few major lineages exhibits two or more oligomeric states. Nearly every protein that has been studied, with variation often existing within and among major lineages. Multimers for these proteins are almost invariably homomeric rather than heteromeric, with subunits encoded by different loci, although there are certainly plenty of the latter for loci in all domains of life. Although there are certainly plenty of the latter for other loci in other domains of life. Okay. Heteromeric. Okay. So almost invariable homomeric rather than heteromeric. That's for these two cycles, the glycolysis and the Krebs cycle. Moreover, in accordance with the broader analysis noted above, there is no association between the level of multimeric complexity and organismal complexity. Yeah, that's funny. For any given protein, the most complex structures are just as likely to be harbored by a prokaryote as a metazoan or land plant. This lack of phylogenetic pattern in protein architectural features is striking when one considers the substantial difference in the population genetic environments, rates of mutation, recombination, and random genetic drift of these major classes of organisms and their likely roles in generating major differences in gene structure and genomic architecture. No, it's not striking, because geometry takes priority. I mean, you make a spark plug basically the same way because it has the same, you know, from your most complex cars today to the ones primitively, there's still a basic architecture in the spark plug. You know, now that we have these cars that have GPS components, this is what I've been trying to hammer the guys, and I don't think it gets through to them. You have a nut and a bolt. Okay? You can have this Formula One car, a Formula One race car. The engines are just unbelievably expensive, like $10 million. $10 million for a car engine. It's very sophisticated. But some of the basic parts are going to be the same. Nuts and bolts, wires. I presume the spark plugs are very similar in architecture. So, you know, then the modern cars have a lot of computerized control, all these sensors for oxygen, etc. And here Michael Lynch is like, this is really amazing. It's been kind of the same throughout all species. It's not like it's gotten very much more complicated. And like, well, why should it? You know, there's an optimal way. Engineers know this. For a given application, there's an optimal way to build it. And if it's a very basic part, it tends to be, you have persistence of how it's constructed. So, yeah, it is a striking feature. And I could have almost totally predicted it, you know. Because the proteins are at a very basal level of how they work. And he's talking about structural geometry. There is an increase in complexification when you go to humans of the same homolog, but that's with the post-translational modifications in the area that they call the disordered, what I prefer to call reconfigurable regions. But this is giving me a lot of food for thought. It's confirming what I have been supposing, and Michael Lynch has spelled it out. Lots of hypotheses, complete lack of empirical evidence. That is evolutionary biology in a nutshell. So I want to thank you all for joining me in this reading of the paper. other people have a different take on this, but it's a great paper, and I'm glad I picked it up. So glory and honor to our Lord and Savior. This is a cross to bear to read through papers like this. It can sometimes be fun, sometimes dreary. This happened to be a halfway fun one. Some of us, myself included, would rather be playing video games and watching sports, but oh well. This is our calling. So let me just close this up, and I'm going to just play some. The usual music. Take care. The Lord richly bless you all.